Chapter 8, the functional synapse. A structural feature between two neurons running in series to each other. This is a fluid-filled space that is the perfect extracellular environment for chemical ligands to be released and transmitted across a physical gap between two cells. The first neuron is called the presynaptic neuron. It conducts electrical impulses in the form of action potentials toward the synapse and to the postsynaptic neuron. The postsynaptic neuron transmits impulses away from the synapse. The focus here in Chapter 8 is the fluid-filled gap in between these two neurons. We will focus on the chemical synapse since it is the most common type that we will run into. The chemical synapse will also be studied when we get into the effectors, which are muscles and glands, in Unit 3. The chemical synapse is governed by the chemical ligands released in the fluid-filled space. On either side of the space, you'll have the presynaptic terminal end feed on one side, shown here coming from above, and the receptor region located on the postsynaptic side, usually the dendrites or the soma, where there are high quantities of membrane proteins integrated into the plasma membrane, acting as receptor sites for the chemical signals to bind. So wherever the synapse is located, the idea is an electrical signal gets converted into a chemical ligand in order to communicate with the postsynaptic side. Remember, electricity can't jump from cell to cell when there's a gap between the two cells. Let's take a closer look at what's going on at the synapse. You have several slides in your notes that show a close-up of what's happening at the chemical synapse. In order for information to transfer from neuron to neuron, the following events must happen in order. These numbered steps start off with the action potentials traveling down the axon to depolarize the plasma membrane at the terminal end foot on the presynaptic neuron. This triggers voltage-operated calcium channels to open in response to this depolarization event at the terminals. The flood of calcium into the ICF compartment triggers exocytosis and the release of the neurotransmitter or ligand. Neurotransmitters across the synaptic cleft will bind to the receptors on the postsynaptic neuron. A response in the postsynaptic cell is induced. It will either be a depolarization event or a hyperpolarization event, depending on the ligand. In this example, we see here on the right a ligand-gated, also called mechanically gated sodium channel. It's bound and goes into the open position, which allows sodium to rush into the cell, causing a depolarization event. Another very important step in this process is the degradation of the neurotransmitters in order to stop the signal. This is labeled as step 6 through 8 in this diagram. The green protein enzymatically breaks down the neurotransmitter signal. Reuptake of the neurotransmitter occurs in step 7 by the presynaptic cell. This recycling step saves time and energy. Notice the vesicular storage, the little purple triangles of neurotransmitter signal already prepackaged and ready to go as soon as we have a depolarization event. In step number 8, we see that some neurotransmitter substances will diffuse out and away from the synaptic cleft, which cleans out the synapse before the next signal comes along. Please include in your study of my full lecture notes the different signal transduction processes with the chemical ligand. You can use figure 8.3 in your textbook to compare slow versus fast cellular processes. Now that we've compared the different operational mechanisms between a G protein operated channel and a fast ligand operated channel, we should also compare what constitutes an inhibitory versus an excitatory synapse. Using 8.4, a fast versus slow excitatory response channel is illustrated for you here. The excitatory synapse will allow sodium to rush into the cell, generating a depolarizing current, making the ICF compartment positive, which causes an increase in membrane potential, referred to as an EPSP, an excitatory postsynaptic potential. Remember, we're talking about two neurons in series. Excitatory postsynaptic potentials are always graded potentials. They can be either fast or slow. The amplitude of depolarization will increase as a result of increased neurotransmitter binding. Depolarizing ion channels cause EPSPs, which are excitatory postsynaptic potentials. At the inhibitory synapse, the postsynaptic cell's membrane potential will move further away from threshold, therefore making the generation of an action potential less likely to occur. Neurotransmitters binding to a receptor at an inhibitory synapse cause the membrane to become more permeable to potassium and chloride ions, causes the charge of the ICF compartment to become more negative, and hyperpolarizing channels, which cause IPSPs or inhibitory postsynaptic potentials, are always going to be inhibitory, going in the more negative than resting position and then moving away from threshold. So you're much less likely to have an action potential generated. With all graded potentials, we are talking about a below threshold event. By definition, that's what a graded potential is, regardless of which direction. So EPSPs do not have thresholds or a refractory period like action potentials do. These small depolarization events are able to have summation, which means they can be added together to get us to threshold level and ultimately fire an action potential. Here in this example, multiple EPSPs occur on a single patch of postsynaptic membrane. Their combined effect is the sum of the individual EPSPs. Threshold is met when we add both of them together and an action potential is fired from the axon hillock. These combined effects is exactly how interpretation of our environment are indicated to the brain. Summation can be either temporal or spatial. Here in figure 8.8, where we see a single neuron synapsing with three different presynaptic neurons labeled A, B, and C. Temporal means depolarizations are arriving close together in time along the x-axis. Spatial summation is when different synapses are simultaneously active. Summation rules still apply, and notice the C neuron is actually contributing to an inhibitory response. But we can still add all of these different activities up to see if we reach their threshold. And we can do this in both cases. The axon hillock is depolarized to threshold and an action potential is fired. Now, please take the time to familiarize yourself with the different types of neurotransmitters covered in this note set. The next portion of this Chapter 8 lecture will cover the nicotinic and muscarinic receptors that bind to acetylcholine, one of our most common chemical ligands.